right, it is a great pleasure to be here. My name is Daniel, and I'm here to present you my work on revealing a cryptic species complex of stony corals from northern Portugal, Dendrophyllia, Scleractinia. Okay, so I'm going to first start explaining what are VMEs, or volatile marine ecosystems. They are ecosystems formed in the deeper parts of the oceans and are typically composed of the oldest metazoan phyla, the porifera, the sponges, and Cnidaria, typically corals. They're known to form dense aggregations, which are considered hotspots for biodiversity. However, they are slow-growing and unfortunately currently susceptible to human impact, such as bottom contacting fishing, pollution, and in the near future, deep-sea mining. So these VMEs can be of several types. They can be coral gardens, coral reefs, sea pen fields, and sponge aggregations. Moving on to the main focus of this presentation, the Dendrophyllia corals. They are stony corals, which are on the order Scleractinia, and they are currently are 31 accepted species. The type species Dendrophyllia, a species described from the Mediterranean Sea. They're usually quite easy to identify because they're very brightly colored, and they're usually yellowish to bright orange. And they also have a particularity, that is that their septa, the skeleton arrangement, is arranged in a Porto-less plan. Basically, what this means is that several septa may fuse with each other along the midway, forming these triangle-like shapes, as you can see on the figure. Last but not least, they're also azooxanthellate, meaning that they do not have the symbiotic relationship with microalgae that you can see on typically on tropical coral reefs. So Dendrophyllia cornigera in specific is a species that was described from the Mediterranean. It is also very characteristic. It is bright yellow, and the colonies are usually arborescent type, although this can vary a bit, and they can be up to 1 meter in height. They are a VME indicator species, usually forming coral gardens, and they can be found from a depth that is very varied, from 55 to almost 1,400 meters. So this work started with an assembly of local ecological knowledge of coral and sponge distributions. This was done through the means of surveys and inquiries to the local fishermen. And with this collaboration, we got a hold of 344 samples that were obtained as bycatch, 5 of which were assigned to Dendrophyllia, which were then analyzed. They had their morphologically analysis with the colony morphology, the general morphology, and the corallite and skeleton morphology. We also did some barcoding with mitochondrial, the CO1, and nuclear ITS, with which we conducted phylogenetic analysis using maximum likelihood and Bayesian inference. And with this, we can discover a separate species of Dendrophyllia, the closest species being Dendrophyllia cornigera, of which we can only distinguish molecularly. So we have a small map of our study area. All of the points are distributed in the northern continental shelf of Portugal. And the material here analyzed were 4 species of what we thought were Dendrophyllia cornigera, but are in fact separate, coming from north of Portugal. And we used material as comparison of 1 Dendrophyllia cornigera coming from south of Portugal, 1 Dendrophyllia from north of Portugal, and 1 leptosomnic privati coming from shallow waters of the Mediterranean Sea. I'm going to show you first the molecular results, which basically kicked off this whole investigation, which was I'm going to say that in accordance to previous studies, the family Dendrophyllidae is a monophyletic family. However, several species are kind of all over the place, and they are probably in need of revision. So I'm going to shift my focus here on the right part of my slide, which shows the focus of this presentation, the Dendrophyllia corals. So the Dendrophyllia corals that we barcoded are in bold, and what we thought were Dendrophyllia cornigera do in fact have some differences. They have 3 base pair differences, which is pretty remarkable considering that this is a very conserved marker for this family. And yeah, as you can see, they are quite different from the remainder of the tree. We do have some substructuring of Dendrophyllia johnsoni and Dendrophyllia cornigera from south of Portugal that we barcoded. from the remainder of the Dendrophyllia cornigera, but simply this is due to the fact that this is a shorter sequence. They are in the rest completely identical. And another thing that quite pops to the eye is the fact that Leptosarcus pravati, a completely separate species of a different genus, has a completely identical sequence with no differences from the Dendrophyllia cornigera. Moving on to the Bayesian analysis, this just has a different, let's say, interpretation of the same sequences. This time we have a polytomy form of the sequences that I mentioned before, as well as one of these Dendrophyllia CF cornigera, because this specimen, the DB42.01, had some degree of interspecific variation, which is also quite unusual for Dendrophyllia corals, in this marker at least. So you can also see the Dendrophyllia CF cornigera, the DB47 and DB80, they form their nice maximally supported clade. Moving on to the other marker, the ITS, we can see here that the Dendrophyllia cornigera is forming, well, a maximally supported clade, this time also with our CF cornigera, although they do have 3 base pair insertions and 1 difference, as well as some degree of variation, interspecific variation, once again with the DB42.01. And also another thing that I would like to point out is that the Leptosarcus pravati on this marker, which are the same individuals that were barcoded for the CO1, this time they have some degree of variation that is enough to distinguish them from Dendrophyllia cornigera, which rules out any possibility that the authors of this sequence might have misidentified these corals. Moving on to the last part of the molecular analysis, the concatenate analysis. Here we can clearly see that what we have with Dendrophyllia CF cornigera is separate from Dendrophyllia cornigera, because Dendrophyllia cornigera from gene bank is forming, and on that same vein, the Leptosarcus pravati are each forming their own maximally supported clades, whereas our CF cornigera are clearly separate, forming a very loose, low supported clade with Dendrophyllia hamea, which is unfortunately its only sequence that we have ITS. For the Bayesian analysis, we can more or less see the same thing. The Dendrophyllia cornigera from gene bank and Leptosarcus pravati are each forming their own maximally supported clade, whereas this Dendrophyllia CF cornigera, the DB80 and DB42.01, are forming a polytomy in the basal part of the tree, showing that they are clearly separate. So we're quite intrigued with these results, the molecular results, so we decided to look a bit further into the morphology. As I mentioned previously, this is a very characteristic coral, and it's pretty easy to identify generally. Basically, when the fishermen catch these species, we can look at them generally and easily assign them to Dendrophyllia cornigera. And in fact, this Dendrophyllia CF cornigera does match completely with the general morphology of Dendrophyllia cornigera. The colonies are quite varied, usually arborescent. In this case, they were quite fragmentary because they were broken off during the fishing process. Their tissues they have their characteristic bright yellow color when alive, and they're somewhat irregular branching with several polyps that can be of different sizes, which is quite different from their closest species, the Dendrophyllia hamea and Leptosarcus pravati. In fact, the Dendrophyllia hamea has a very uniform polyps. They're all of more or less of the same size and the same height, usually quite short. They're organized quite nicely within the branches. On the other hand, Leptosarcus pravati is a solitary species. It does not form colony, which is a key characteristic of all Dendrophyllia species. So we decided to look a bit further. Now we are looking at the skeleton of the polyps. First, I'm going to show the easiest one to distinguish, the Leptosarcus pravati, because it does not form the typical Portugal plan that all Dendrophyllia corals have. To give you a brief reminder, this means that the septal arrangement forms some certain fusions, forming the triangle-like shapes that you can see here on the left, if you can see my cursor, which you can clearly not see with Leptosarcus pravati, ruling out the possibility that this is a Leptosarcus. Moving on to its another close species, the Dendrophyllia hamea, which also occurs on the same location. This coral, as I mentioned previously, has smaller polyps, resulting also in a much smaller corallite, and also in fewer septa. The Dendrophyllia hamea usually has around 48 septa, usually in multiples of 6. However, Dendrophyllia CF cornigera and also cornigera can easily exceed 70 septa, which is quite different from Dendrophyllia hamea. So moving on to the last comparison that we made with Dendrophyllia cornigera, the true cornigera. So here we had a lot of trouble trying to distinguish these two because there's not really any clear differences. As much as we looked at different polyps, we might see some irregularities in Dendrophyllia cornigera. You can see here, just to make it a bit more simple, like on these triangles that you can see on the cursor, you can see some new septa forming, which is quite irregular, but this might be due to the fact that this coral was probably growing when we collected it. So we definitely need a bit more individuals to maybe try to investigate this more. So to finish off my presentation, I hope I'm on time. But based on molecular data, there is probably a cryptic species complex of Dendrophyllia cornigera occurring on northern Portugal, because we simply cannot so far distinguish it morphologically, although we are still in the process of trying to find more characteristics that might distinguish these two. So in order to do that, some future considerations that are in the works. We do have some scanning electron microscopy images of the sea of cornigera, but we are still in the process of getting them from the Dendrophyllia cornigera, and we are hoping that there might be something that we just simply missed when looking at the stereo microscope. Another thing that is quite interesting and is a study that is really like outside of the box is the analysis of the Cnidaria, the stinging cells of corals, which is something that is rarely done with Scleractinia corals, but it has been done with Dendrophyllids, and it is something that we are definitely looking forward, and we are definitely going to try to do in order to maybe try to properly describe this species of coral. And the last small closing off remark is that the only Dendrophyllid corals that are listed as vulnerable marine ecosystem indicator species are Dendrophyllia cornigera and Dendrophyllia hamea due to their ecological importance because they form coral gardens. However, is this Dendrophyllia sea of cornigera is shown and properly described, and this list might be in need of reconsideration, and these species might have to be included in that list as well. So that's it from me. I hope you guys enjoyed my presentation. Thank you.